Look closely at a Drosophila fruit fly. Behind its red eyes and delicate wings lies a biological instruction manual written in billions of chemical letters. This is a DNA sequencing chromatogram, representing a tiny fragment of raw genetic data. When we sequence an entire genome, we generate billions of these individual nucleotide reads in a single run. Because of high-throughput technology, repositories like GenBank are continuously flooded with an unrelenting stream of genomic, transcriptomic, and proteomic sequences from thousands of different species. It is physically impossible for a human being to manually read through this constant influx of A's, T's, C's, and G's. With global databases now containing hundreds of billions of nucleotides, the primary challenge has shifted from simply collecting the code to designing the systematic queries required to understand it. That is why modern repositories do not store sequences as giant scrolling text files. Instead, they organize this information into relational databases. A relational database structures data into strict interconnected tables. Each table breaks down complex biological entities into specific fields, like an organism's species, the sequence string, or its accession number. To navigate these fields, bioinformaticians use SQL, or structured query language. SQL provides the structural vocabulary needed to perform these lookups across massive datasets, allowing a researcher to retrieve specific, functional insights from an archive of millions of entries. Building a SQL query starts with targeting the exact location of the information you want, using two foundational commands, select and from. The from command points the system to the correct table, like a master list of proteins. Then, the select command extracts only the specific columns you need from that table, while the rest fade into the background. Even after isolating a single column, you are still looking at millions of rows of data spanning thousands of different organisms. To refine this list, researchers use the where clause. This highly specific biological filter commands the database to return only sequences meeting exact conditions. For example, it instantly filters a global protein dataset down to sequences from Drosophila melanogaster. Select, from, and where provide the foundational syntax to reduce a global archive of millions down to the specific, actionable set of sequences relevant to a single experiment. Isolating data is only the first step. Biological systems are heavily interconnected, but large databases often store information in isolation, keeping raw DNA sequences in one table and the resulting protein structures in an entirely different one. To map relationships, bioinformaticians rely on the join command. It identifies a unique overlapping identifier, like an accession number, existing in both tables. It uses that shared ID as a hinge to fuse the disparate datasets into a single matrix. Once those connections are made, researchers need to identify overarching patterns within the merged data using the group by and order by commands. These commands group similar records together, allowing biologists to aggregate metrics, like calculating the average expression levels across an entire gene family, or sorting those results to find the most highly conserved segments. By combining join with these aggregation tools, a researcher can transform thousands of scattered data points into a cohesive statistical understanding of a complex biological system. This interface shows the NTRES protein database from the National Center for Biotechnology Information. It is exactly the kind of portal where everyday bioinformatic queries take place. Whenever you type a gene name or an organism into one of these public search boxes, you are actually triggering a complex SQL query to run behind the scenes. Advanced bioinformaticians take this a step further, integrating SQL directly with programming languages like Python to automate large-scale tasks like RNA sequencing analysis or variant calling across entire populations. Writing these queries requires a firm grasp of computer science syntax, but the ultimate goal is deeply biological. Constructing a precise SQL statement is the modern equivalent of peering through a microscope. It is how today's scientists parse the noise, discover new mechanisms, and command the world's rapidly expanding library.